Okay. And I would say for the rest of you, like we can keep this pretty conversational. So like if you have a question, you can just sort of chime in because I think right, part of the goal is for all of you to not only learn the thing that you're sort of presenting on, but like everybody's, right? And I think they're all like really good topics. Anyway, go ahead, Naya.
Okay, so my topic is on PROTACs, and that stands for proteolysis targeting chimeras. So what are PROTACs? Well, it's a new type of medicinal technology, and PROTACs themselves are heterobiofunctional molecules, meaning that they have two binding sites. And essentially what they do is they initiate intracellular proteolysis, which describes the breaking down of proteins into smaller peptides and amino acids. And so there are two different protein binding sites, and one of the binding sites is responsible for attaching the E3 ligase, which I will go into further.
Use your
And
laser
then,
pointer.
what was that?
Use your laser pointer.
Oh, sorry. The E3 ligase, which is like the ligand for the E3 ligase, so like the connecting point. And then the other binding site, it acts by, it's where the targeting protein is supposed to attach to the PROTAC molecule. So why PROTACs can be useful? Well, naturally proteins do need to degrade. So if there's a misfolded protein, a protein with a mutation, or proteins that just aren't needed anymore, they have a natural protein degradation system that they go through. So essentially what PROTACs do, which I'll kind of go into further later when I talk about more of the medicine part of it, like how PROTACs are used as drug therapies, it basically takes advantage of the fact that proteins naturally degrade. And it's particularly helpful in cancer, defense against cancer, or cancer medication, because it promotes apoptosis, which is cell death. And that's a crucial system in place that helps to, I guess, diminish the cancer cells, tumor cells, and so some of the, I guess, proteins that PROTACs can target are transcription factors, skeleton proteins, enzymes. PROTAC needs to be successful. Well, it needs the ubiquitin proteasome system, which I will go into further, and that's the system that it kind of hijacks. And then the three parts of that system are the E1 activating enzyme, the E3 ligase, which is also extremely important. Kind of how I mentioned earlier, it needs the E3 ligase in order as one of the, I guess, at one of the binding sites. So the ubiquitin proteasome system, also known as the UPS. Well, first I'm gonna define a few terms just so they're familiar. Ubiquitin is a small protein that is found in almost all cellular tissues in eukaryotes, and it helps carry out some normal cell processes. Ubiquitination is kind of the inactivation of proteins with ubiquitin attached to it. And the proteasome, okay, so here's what a ubiquitin might look like. And then it goes through more, I guess, biological processes through these three different phases. I think I would describe them as. So I'll describe what the proteasome is. So the proteasome, it's a protein complexes that are responsible for degrading misfolded, mutated, or proteins or proteins that just aren't needed anymore. So the three parts of this system are, well, first you have the E1, which is the activating enzyme. So that's just the initial step in ubiquitination. Then you have the E2, which is it's the conjugating enzyme. So it connects E1 to the target protein to start the process of degradation by the proteasome. And then you have the E3 ligase portion, and that's well, ligases, I guess, in general, are enzymes that can catalyze the joining of two large molecules by forming a new chemical bond. So in this case, what an E3 ligase does is I guess, it starts the process of the degradation, like how that protein is going to degrade. So the E3 ligase is probably.
Can I interrupt you? Oh, I'm sorry. My voice sounds weird. Can I just sort of emphasize one thing that Naya said that's very important, is that the attachment of that small ubiquitin protein to another protein is what tags it to be destroyed. So that's sort of like what the whole machinery is meant to do, is attach a small protein to another protein to make that other protein get degraded.
Mm-hmm. So the E3 ligase, well, it's a natural protein disposal system that's found in proteins. And what happens is that the E3 ligase will identify mutated or misfolded proteins, or as I said, proteins that aren't needed anymore. And it tags them with the ubiquitin protein, which guides that target protein. So I guess what I mean by target protein is the protein that you're selecting, the protein that's causing a problem, the protein that's it's just not needed anymore. And it tags that protein and it guides the target protein to the proteasome, which will then degrade the protein into small peptides or amino acids. So how PROTACs interact with the UPS? Well, as I kind of mentioned earlier, it hijacks the UPS. So it I guess takes over that natural system that's already in place to degrade proteins. And what it does is it kind of summons the E3 ligase to tag the target protein, the selected protein, the protein you're trying to get rid of. And so it can be recognized for ubiquitination and degradation by the proteasome. So PROTAC is pretty unique because it doesn't act as an ordinary enzyme inhibitor. Enzyme inhibitors they pair with an enzyme to prevent normal substrate enzyme combination and the catalytic reaction. But what PROTACs do is they do
Uh-oh. Is anybody else up there? I think Naya's screen got frozen. Oh, no. Thanks, Michael. Uh-oh, she's gone. There she is now.
Oh, sorry about that.
No problem.
Okay, can you hear me?
Yep.
So what did you guys miss?
Can you share screen again? Oh, I might need to remake you. Oh, you got it. Good. Okay, good. Let's see.
Okay, so I talked about.
It was basically the beginning of this slide.
Okay. So I mentioned it's not an ordinary enzyme inhibitor. It takes advantage of the normal process of a protein. One of their common ways that for cell maintenance or protein maintenance. And a lot of cancer research, some of the therapies for it that they've been focusing on are antibodies, siRNA, and small molecules, which is where PROTAC kind of fits into the mix. And one of the goals of drug therapies, what they, I guess, were looking
Whoops. Oh no. Sorry.
Can you see that?
Yeah.
Yeah.
Okay. So the BRD4 protein. So essentially what it is, it's a domain, an extra-terminal domain epigenic reader protein. And this is a protein that's commonly used to
Oh no, this is not good. I don't know what to do here. I'll pause the recording.
Okay. Can you see this?
Yes.
Okay. So I kind of just wanted to recap what ProTexts are, because I think it's important to know how ProTexts work because of the different systems that are in place, and it can be, I guess, difficult to understand if you don't know what the I guess the system already does. So ProTexts are molecules that induce selective intercellular proteolysis, and it uses the ubiquitin proteasome system in proteins to degrade these proteins, that you are target protein, something that's misfolded, mutated, or something you don't need anymore. And does that by summoning the EPase and attaches ubiquitin to target protein, and then sends the proteasome system to further read it. And that's all.
Nice, Naya. Sorry, that was your voice got a little garbled, at least from my end. I don't know, could the rest of you kind of hear a little bit? Yeah. Can you go to slide 9 real quick, Naya?
Sure.
Just to point out, I think and actually it might help if you not share screen, but just leave it like, it probably doesn't matter. Just to show people, like maybe for what the proteins are on this slide, and then what the small molecules are, and then specifically the ProTac. Are you there, Naya?
Sorry.
I'm sorry.
Yes, I. Okay. So you want me to not share the screen?
Yeah, your audio is really bad right now. Can you hide your video? That sometimes helps. Just like stop your video. I don't think she's. This is actually a pretty key slide here too. Can you all still see it? Yeah, that I'll just sort of point out that the purple protein is the E3 ligase that directs ubiquitination, and the yellow protein is the protein that they want to degrade. So, and underneath each of them is a molecule that binds to each protein. And all the PROTAC is, that's in the middle. It's those two binding partners with a linker. So what the PROTAC does is it brings the E3 ligase right next to the target protein to enable that ubiquitin transfer reaction, and then the protein gets destroyed. This is actually, I'm sorry that Naya, your audio's not, it's just not working. But actually, Naya.
Oh my goodness, this is really bad. It's sweating.
It's Naya, there's two of you now.
Wait, hold on. I'm like, this is just not okay. Wait, so what did you just say?
I think Scott's frozen.
Yeah, now Scott's frozen. Naya, there's two of you too.
I know. I'm
Do you
sweating.
see that?
My computer's not working.
Sorry.
And I don't know how to, I'm completely exited out of this thing, so I don't even know how my screen is still up there.
A ghost. It's so cool.
Bye, Scott.
Scott's gone.
This is still recording.
Hold on, you're gonna have to wait a second.
Who's beeping?
You guys
Oh,
hear?
sorry.
Oh.
There's an alarm going off.
I thought that was my neighbor's house.
Wait, so the PROTAC is just the stuff in red? Is that what he was saying?
My
No,
computer and the
it's
internet
all
connection.
of
That's
it.
what happened.
It's all of it?
Yeah, so it's the one that is able to bind to the purple and the one that's able to bind to the yellow connected by a linker. That way the two proteins like stick together.
Oh, I thought
It
the
forces them to like join.
E3 ligand, I thought that was the same as the purple one.
The,
what I'm talking
the
about?
purple protein is a ligase. The ligand is the thing that bonds to it.
They look exactly the same. Oh boy.
So do you guys understand PROTACs?
Yeah.
Or what they do?
100%. It was really good presentation, Naya. Like, aside from the technical
Yeah.
difficulties, that was really good.
Remember when your voice like turned to Darth Vader halfway
Oh,
through?
that was brutal. You don't even understand, every part of me is sweating right now.
At least it's just us and Scott.
Where is Scott?
Yeah, wherever he is.
Scott just like disappeared.
Yeah. Does someone have to like let him in?
No, because it's his meeting.
He should be the host, so it should be like
Naya, you're the host.
whatever.
You're the host.
I know.
Oh. So if you see someone in like a waiting room, it's probably Scott.
This was all a plan to take over.
Naya,
Wait, Emily,
original
how
you
long-
is gone.
Good.
What?
How long is yours, Emily? Like, I'm just wondering how many slides it should be because I have no idea. Mine's like 15.
Yeah, mine's like, I can't really say because sometimes the same slide is duplicated, but it's just like I added a little more text. So there's like
Oh, okay.
20 some, but it's really not 20.
Oh, gotcha.
Mine has a lot of slides.
Wait, the guidelines he was talking about, was that like the little PowerPoint he added to Sakai with all of our synthesis in it?
No, it was like a Word document. I
Oh,
can't
where
remember
is
where I found it. It was
I can't
just.
really navigate the Sakai site.
I have no idea. I just click on things until I find stuff.
I don't
Yeah.
think it's in Sakai. I think it's whatever he sent us in that Memorial Day email.
Yeah. It
Oh,
was just like the one for
Sakai.
this presentation. It's just like a list of like bullet points and stuff.
okay.
Oh my god.
Sorry, I actually got kicked out too. Something. Sorry about that. Okay. Does anybody have any questions for Naya? Does everybody get the basic idea of how ProTax work? And of course, she'll talk about them more and some specific examples as time goes on. Thanks, Naya.
Mhm.
Did you watch the video on the Arvinus or whatever the company's name is, their website?
I did. I know that was really good at explaining kind of how it works.
I thought so too.
I looked at one of the, I guess, ways that I understood, I guess, what an E3 ligase was. Underneath the video, they had a description of, how it kind of works, a breakdown of basically, I guess, what the video was talking about. And so that was helpful.
Good. That's good. Cool. The one thing I think too that I know it will happen to you soon, and actually with other people too, but it's sort of the point of these as we get into sort of reading results that people have published in papers, is being able to interpret the data that you're looking at in those papers. So, so for you, and actually for most people on the second day, I'll want you to show and explain certain data from certain papers. And that is going to be very difficult for many of you, I think, in the beginning. But it won't be like that forever. So, so maybe it is actually a good idea to have a little bit of a delay between the introduction to your topic and then sort of the first results-based paper from your topic. You follow my meaning? Because you'll be looking at a lot of gels and different kinds of experiments that you're probably just not super used to doing. All right.